The slides should be visible to you as I can see it, but there is no confirmation yet whether you can see the slides. So anyway, let us start and see if others join. They can join in this meantime. So we will start with our week 7 questions. The questions that were related to week 7 lecture materials, and then I will move to the week 8 lecture materials. In the last session, we discussed some of the questions. I think 5 of the questions were discussed, and we will start from question number 6. Yes, after question 5 we solved in last class. Today we will begin from question number 6. So let us start. This is the first question. It states, choose the fatty acid in each pair that has the higher melting temperature. These are the fatty acids that are given, P, Q and R. These are the three sets. One second. So this one is your first set, it is your second set, and this is your third set. In each of this set there are 2 amino acids. You have to select in the first set out of these 2 amino acids which will have the higher melting temperature. These are your options. If you can see the question and hear my voice, then you can try to solve the question and write your answer in the chat box. Otherwise, I will directly start discussing. Okay. There is no reply yet. So let us discuss this question. So first of all, we can clearly see this is there are generally 2 ways we can designate the different kind of fatty acids: delta designation and N designation. Now in this particular question, the designation that is mentioned is delta designation, because from this you can clearly see this is represented in delta designation. So in case of this first set, these are the 2 fatty acids. What does this delta designation mean? That we need to know. So it is written like 18, 1, delta 9. So what does this number represents? In the last session as well, I mentioned it that the first number, it tells you how many carbon atoms are there. The next number tells you how many double bonds are there. And this number, which is to the power of delta, this tells you the place of the double bond. 9 means the double bond that is there is between carbon 9 and carbon 10. Okay. So the very first number tells you about the total number of carbon atoms. The next number tells you about how many double bonds are present, and this third number tells you that what is the position of this double bond. So we already deduced that for the first, in case of the first set, this amino acid contains 18 carbon atoms, 1 double bond which is placed at between C9 and C10. Now what about the second one? In this set, the second fatty acid, if I write it down, its delta designation is written as 912. That means here also we have 18 number of carbon atoms, same as the first one. How many double bonds are there? There are now, from this number we can tell, double bonds, there are 2 double bonds. And what is their position? 9 and 12. That means one of the double bond is present between C9 and C10. Another one is between C11 and C12. Fine. Now we all know how many number of carbon atoms are there, how many double bonds are there, etc. How can you relate this to the melting temperature of the fatty acids? So if you remember your lecture materials or the lecture session, then it is clearly told that what is the general structure of a fatty acid? You have your COOH group and then a long hydrocarbon chain. The more you put double bonds here, that means you are putting more kink to the structure. How do you do it? Suppose your carbon chain or hydrophobic carbon chain looks like this. In another case, I am not writing the COOH anymore here. In another case, your carbon chain looks like this. So whenever you place a double bond here, that means you are adding a kink to your system. Ultimately, this whenever you will form a gel or crystalline these fatty acids, what will happen? If there is no double bond, they will very easily be placed in a order state like that. It is very easy to place them or keep them in a order state. But whenever there is a double bond, that means you are hampering the order that was present, especially because of the cis conformation of the double bonds. Now the way I have written here is in the trans conformation, but this can similarly exist in cis conformation as well. In that case, it will look something like this. Whenever you are putting this any kind of double bond, the order that was initially present is no longer valid. You can clearly see that after this formation, we will not be able to put these chains one upon another in a proper arrangement or a proper crystalline state. This means the structures which does not have double bonds, they are highly arranged, or we can say highly or regular arrangements can be achieved for them. But whenever there are double bond, the arrangement is very less. How will that impact the melting temperature? You all know, whenever something is very nicely arranged like crystal, in case of like crystalline arrangements, the melting point goes high. Because melting points means what? You are basically going from a crystalline, highly crystalline state to a non-crystalline state. Or in case of fatty acid, you are going from a gel-like state to a liquid crystalline state. That means you are decreasing the crystallinity that is present in the system. So in case of highly arranged system, if it is packed very nicely, then you will require more temperature to break that regular arrangement and take it to some irregular kind of conformation. The more the crystallinity, the more temperature you will be needing to transform it to a non-crystalline state. That means whenever there is a fatty acid chain that does not have a double bond is compared with another fatty acid with same number of carbon atom but has one or two double bonds, that means you are adding kinks, thereby hampering the crystallinity in the system. Therefore, the melting temperature that you will need will also become less. Fine. So in the first case, we can clearly see for this one we had only one double bond, but in the second case we had two double bonds. That means where is the kink is higher? In the second case, as there is number of double bond is higher, in that case the kink is also higher. That means the structure is much more irregular in this case, which again means for this particular fatty acid, you will require less temperature to take it to a non-crystalline state. But in this one, you will require higher temperature to take it to a non-crystalline state. That means which one has a higher melting point for our set 1 is the number 1 fatty acid. This one has the higher melting temperature. Moving to the second one, that is Q. Again, the designation is delta. For the first one, it is written as 18,0. This again means that there are 18 carbon atoms, but there is 0 double bonds, right? But for the second one in this set, it again has 18 carbon atom, but has 1 double bond. Just like we did in set P, in this case as well, wherever there is more number of double bonds, that has lower melting temperature. That means wherever there is less number of double bonds, will have higher melting temperature. So in this case, this has no double bonds. That means higher crystallinity. That means higher melting temperature. So for set Q, this one will have higher melting temperature. Moving to the set R, the first one is written as 18,0. The next one is written as 16,0. Now in both the cases, from this number 0, we can clearly say that in both the cases, there are no double bond present. In both the cases, there are 0 double bonds. But how they differ? They differ in the total number of carbon atoms. In the first case, you have 18 carbon atoms, whereas in the second case, you have 16 carbon atoms. Fine. So how it will affect your melting temperature? Suppose, let me draw it like this. This has 1, 2, 3. Suppose your fatty acids look something like this. I'm just giving you an example. One has less number of carbon atoms and another one has more number of carbon atoms. Now, whenever you will try to arrange them, obviously, if you try stacking them, here you will have much more ordered structure than this one. That means, if you increase the chain length, it will give you more crystallinity in the structure, or the gel form will be more ordered. The higher the chain length, more will be the ordered structure. And whenever the structure is more ordered, you will require higher temperature to melt it to a non-crystalline state. That means between these two, as the first one has more number of carbon atoms, this will have higher crystallinity followed by higher melting temperature, and the second one, as it has less number of carbon atoms, it will have lower crystallinity followed by low melting temperature. So now we all have that among these three sets which will have the higher melting temperature. For P, it should be 18, 1, delta 9. Okay. For Q, it should be 18,0. For R, also it should be 18,0. The only option that matches with our deduction is option number A. For P, 18, 1, delta 9 is highest. For Q, 18 is highest. For R, also 18 is highest. Okay. So, hope this is clear. Moving to our next question. Here it states, the cholesterol will insert into the bilayer membrane with its, there is a blank which is denoted as W, oriented towards the, again there is a blank, and the third blank is would adjacent to the phospholipids. So these are your options. You have to fill W, X, Y and Z. This is your full sentence. You have to complete this sentence by putting appropriate W, X, Y and Z. Fine. So if you do remember the structure of cholesterol, one second, let me write it down. The basic structure for cholesterol. Sorry. So this is a generalized structure for your cholesterol. Now it is saying the cholesterol is getting inserted into the bilayer membrane. So how does the bilayer membrane looks? It looks like this. Where these are polar head groups. These round ones are polar head groups. And this intermediate region is your non-polar part. So now if cholesterol wants to insert into this kind of bilayer membrane, how should it orient itself? First of all, you can see in cholesterol structure there is a hydroxyl group. That means this particular functionality or OH functionality, it is a polar group. So obviously it will try to orient it in such a way that it remains in a contact with another polar region, right? So in case of cholesterol, its hydroxyl group, the first option is hydroxyl group, you can see. The hydroxyl group will try to orient towards the aqueous phase. So if you look at it, this portion is aqueous phase, this portion is aqueous phase, this is your hydrophobic phase. So if you try to put cholesterol here, this OH moiety will try to orient itself towards the aqueous phase, which is polar, and its hydrophobic ring system. These fused ring parts are fully hydrophobic, so they would try to remain adjacent to the fatty acid chains. Obviously, if these are your fatty acid chains, this is due to the hydrophobicity of the fatty acid chains. Now if you are putting cholesterol here, this non-polar part would try to stabilize itself by embedding itself adjacent to the fatty acid chains of the phospholipid, which is basically the hydrophobic parts of the phospholipid. So what should be the correct option after discussing all these things? Option number 1. Now let us read the sentence again fully. The cholesterol will insert into the bilayer membrane with its hydroxyl group, W equals to hydroxyl group, oriented towards the aqueous phase, obviously just as we discussed, and the hydrophobic ring system, that is this fused ring system, would be adjacent to the fatty acid chains of phospholipids. Obviously, just as we discussed, now this whole sentence makes sense. You just need to remember what is the polar and non-polar part for phospholipids or bilayer membrane and what is the polar and non-polar part for cholesterol. And then you know that polar will go towards the polar part of another molecule and non-polar will go to the non-polar part of another molecule or another system. Let us move to the next question. It states the fluidity of a lipid bilayer will be increased under which condition? Increased when? What? So these are your 4 options. So first of all, you need to remember or know that what is meant by the fluidity of lipid bilayers. If any of you know the answer, you are always welcome to write your answer in the chat box. I'm constantly reading it, so you can definitely write. If you know the answer, you can write it in the chat box, okay, and then can check with my answer later. So first of all, let's go back to the question. So first of all, you need to know this particular term fluidity that is mentioned here. So how does lipid bilayer looks? Let me write it down, draw it a bit properly. So this is how a lipid bilayer looks. Fine. These are your polar head groups. These round ones are your polar head groups, and these are your hydrophobic fatty acid tails. Fine. Now what we are meaning by fluidity? This particular conformation, this bilayer conformation, can exist in two ways. One is highly arranged gel state. Already from the picture you can assume or you can see that this is highly ordered or crystalline kind of state. Everything is pretty much ordered. So this is your gel state. What can happen? It can also exist in a randomly oriented state, like will kind of look like this, which is known as liquid crystalline state. Now the question is, which one is more fluid? If you think properly, if there is a very good arrangement in this case, now you want to put something from outside, go through this layer and take it inside the cell. Suppose this is your intracellular part and this is your extracellular part. Now you want to take something from outside through this bilayer membrane to the inner side of the cell. But the problem is, if the bilayers are very much orderly arranged and highly crystalline, then obviously the transport will be hampered, because just think about, like say you have a stack of books. Suppose you have arranged them in proper order. Now you take a pen or pencil and you want to push it through that stack of books. Will you be able to do that? No. If the books are properly stacked, then there are no place left for you to put anything from outside and take it to the other side. But if someone placed the books, suppose, in a random nature, not in a properly stacked way, but randomly scattered, then there will be many holes and channels and everything through which you can put your pencil and take it to the other side. Like, same thing happens in this case of lipid bilayers as well. If it gets distorted, then there are some channels through which other molecules can actually pass. This is called the fluidity of bilayer membrane. You need some kind of randomness in your lipid bilayer so that other molecules can have some passage through it. Otherwise, if it is fully ordered and crystalline, like in this gel form, then there will be no transport through the membranes. Then the whole system, the whole biological system will fail. So this transformation from extremely ordered to liquid crystalline state, from gel to liquid crystalline state, and the generation of these passages is actually known as the fluidity. That means this liquid crystalline state is much more fluid now, much more fluid now, because you can pass more molecules or more substances from outside to inside for transportation. Now let us get back to the question. It is asking that fluidity of a lipid bilayer will be increased when. So now we all know that this is our fluid state. To get to this state, you have to increase the randomness in this bilayer membrane. How can we do that? This is your correct answer. Why? Because just in the previous question we discussed that the longer the fatty acid you take for making this bilayer membrane, they will be more properly arranged in a crystalline order. The shorter the chain you take, the less crystallinity would be there. So if you decrease your chain length, more will be the randomness, that will lead to more fluidity. That means decrease in chain length helps in getting fluidity. The next part is degree of unsaturation. Again, as we discussed previously, that when there is no unsaturation, this is a fully saturated system, there is no double bond. In those cases, the packing is very ordered, or in a crystalline packing we can say. But whenever you put a double bond here, it will also take a cis conformation that will look something like this. That will look something like this, which will put a kink to your system, and you will no longer will be able to arrange them in a properly ordered crystalline state. That means you can increase you can decrease random just a bit, sorry, I was getting confused. Okay, so you can actually increase the randomness that is present by introducing more number of double bonds, thereby introducing more number of kinks. That means if you increase the unsaturation, which is directly means if you increase the number of double bonds, that means there will be more kink to your system, and that will be less ordered. This means the fluidity will again increase. Okay, hope this is clear. Moving to the next one. This is your structure. Given the structure of this hypothetical fatty acid, the correct nomenclature of the given structure would be what? First of all, there is a mistake. In all fatty acid, this should be COOH. Okay, this is your proper structure. So this is the structure of a hypothetical fatty acid. You have to identify its correct nomenclature. From the options you can already see we are using delta designation. I already discussed about delta designation, but again, in case of delta designation, where does the numbering start? It starts from the site of the COOH group. In case of N designation, we start from opposite of the COOH group, but in case of delta designation, we start from COOH group. So let us mark the carbon atom first. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So we have marked it. How many carbon atoms are there? 20. So in case of delta designation, what do we write first? Total number of carbon atoms. Fine. So that is 20. After that, what we write? We write how many double bonds are present in this fatty acid. So let us look how many double bonds are there. 1, 2, 3, 4, 5. So there are total 5 double bonds. So after 20, then we will write what? That there is 5 double bonds. Now in delta, we can talk about the position of these double bonds. So what are their positions? One is between C5 and C6. So we can write it like 5. The next double bond is between C8 and C9. So we can write it as 8. The next double bond is between 12 and 13. That we can write as 12. Next one is 15 and 16. So we can write it as 15. And the last one is 18 and 19. We can write it down as 18. So this should be your delta designation. I think the correct option should have been, okay, there is a mistake in this position. It is mistakenly written as 16. It should be 15. Okay, yes. So this is the correct designation or the correct option. 25, delta, 5, 8, 12, 15, 18. Okay, so that is how you specify the nomenclature of a fatty acid through delta designation. Okay, let us move to the next one. Which of the following acts as a barrier to most water soluble substances? First option is phospholipids, second option is hydrophilic heads, third is nucleus, and D is deoxyribonucleic acid. This particular question is very easy. We all know among all the options, only phospholipids, they have these hydrophobic tail parts and hydrophilic polar head groups, which they can use to form a bilayer membrane, which gives you, so this part is hydrophilic, but this intermediate region is your hydrophobic. One second. Hydrophobic. Now because of that, whenever you put some water soluble substance and you want to take it through this membrane, it will not be able to pass easily. It will hinder because inside of this membrane is hydrophobic. So any water soluble substance, it is water soluble means it is a hydrophilic substance. So it can come up to the polar head groups, but whenever it will enter inside, it will get a hydrophobic region which will not be good for its solubility or its stability. So it will not be able to transport through. So out of all these options, only phospholipids, that is basically your bilayer membranes, they do act as barriers for most water soluble substances. So the correct option for that should be option number A. Fine. This is your next question. This is all you know, just straight taken from the lecture material. Plants and fungi generally do not contain cholesterol. Cholesterol is a sterol that is named, that is found in case of for animals. So that particular sterol we call as cholesterols. But in case of plants and fungi, we also can get other types of sterols. And what is their name? That is being asked in this question. So in plants, we call them, what do we call them? we call them stigmasterol, and in case of fungi we call them ergosterol. So in animals we call it cholesterol, for plants that is called stigmasterols, and for fungi they are ergosterols. Okay, moving to this next one. Again, this one is very interesting. The question states palmitic acid and palmitoleic acid are two fatty acids whose structures are given provided are the melting curves for the two given molecules. Assign the curves as that for palmitic acid and palmitoleic acid. Okay. So this is the melting curve. This is one melting curve, this blue one, and the red one is another melting curve. Okay. X will be either of these two structures. Similarly, Y will be either of these two structures. You have to identify which one among these two structures, palmitic acid and palmitoleic acid, which one is X and which one is Y. You just need to identify it. Okay. How can we do that? We know the difference comes from the melting temperature. For X, we can see the melting temperature is very low, 0.5 degrees Celsius. Whereas in case of this red one or Y, the melting temperature is 63 degrees centigrade. So what does that mean? If a fatty acid does have a very low melting temperature, that means, so what is your melting temperature? You are taking, you are moving from a crystalline state to a random state. If the melting temperature is low, that means this crystalline state that existed initially was not very ordered. That is why to take it to a random state, you do not need very large amount of heat. Very easily, by putting very small amount of heat, you can take it to its unordered or random structure. But whenever the melting temperature is high, that means the crystallinity that existed was very good in the system or very high. That is why when you want to take them to a randomly oriented state, you need to give higher amount of heat to that particular fatty acid or that particular system. Okay. So obviously this means our X was not very ordered initially. The crystallinity is less, whereas this Y1, this was very ordered. That much we have already configured for X and Y. Now let us see at the structures and then deduce which will have more ordered and which will have less ordered structure. Now for palmitic acid, you can see, first of all let us look at the number of carbon atoms. How many carbon atoms are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Now in case of palmitoleic acid as well, we have, sorry, I missed the COOH carbon here. Okay, so just apart from COOH, I was just marking 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So apart from the COOH group, both of these fatty acids does have same number of carbon atoms. So in basis of carbon atoms, we would not be able to deduce that which one is going to be more ordered and which one is going to be less ordered. But if you look in case of palmitic acid, it has no double bonds. That means there is no kink present in the structure. It can very nicely arrange this hydrophobic chain one after another, which can lead to high crystallinity. But in case of palmitoleic acid, there is a double bond, and due to the cis configuration of this double bond, there is a kink in the structure. In this case, the fatty acids look like this. But due to this kink, here the structure will look something like this. That means whenever you will try to arrange these fatty acids in the bilipid membrane, it will not be properly arranged. That means the crystallinity would be very much less. Now that we have this answer, let us match with the graph. So palmitoleic acid was not very ordered. That's why the melting temperature of it was low, whereas palmitic acid, there was no kink presence. It was highly ordered. That means it has high melting temperature. So our X is palmitoleic acid and our Y is palmitic acid. Okay, hope all of you understood. Moving to the next one. A cerebrocyte, question number 13, it states, a cerebrocyte is a ceramide with a blank is there, such as glucose or galactose as the polar head groups, whereas gangliocyte is a ceramide with a polar head group that include acidic sugar activating salicylic acid. These cerebrocytes and gangliocytes are collectively known as what? So first of all, what we need to remember, that what was ceramide, gangliocyte and cerebrocytes. So first of all, if you do remember the classification of fatty acids, they are based in what kind of monomeric or basic unit you are using, whether you are using glycerol or you are using sphingosine. Based on that, the initial classification was made. If the mother body is glycerol, then those are one second. Then those are called glycerophospholipids. But if they contain sphingosine, then they were called sphingolipids. These were the basic division of phospholipid based on their structures. Now ceramides are another classification that comes for sphingolipids. So we are now not bothered with glycerophospholipids. For now we are not bothered with it. We can further classify sphingolipids to different types. First of all, what will be ceramide? If you remember the structure of ceramide, then there is a NH3 plus group, there is a long hydrocarbon chain, and there is a OH group. So we have our NH3 plus group, have our OH group, and a long hydrophobic carbon chain. Now, if you attach a fatty acid here through the formation of a amide bond and the other parts are free, only a amide bond has formed here with the NH2 and another COOH of a fatty acid through the amide bond formation, then we call them ceramide. Fine. Now what will be the next classification? So we have came up to ceramide. Now we can see there is still another group free in case of sphingosine, that is this particular OH group. Now this OH group can be attached with a sugar molecule. Fine. sugar molecules have different COOH groups. This OH can attach to a sugar molecule. Depending on what type of sugar it is attaching, we can further classify it as cerebroside or ganglioside. If the sugar that you are attaching is a monosaccharide, then we will call them cerebroside. If the sugar that you are attaching, if it is a complex saccharide, then we will call them ganglioside. But in both the cases, as we are using sugar moiety to attach with this OH group, they are collectively called as glycosphingolipids or glycolipids or glycosphingolipids. Okay, glycosphingolipids. Sorry for the handwriting. Okay, so you need to remember this total classification, that whenever we are adding a sugar moiety with ceramide, we call them glycosphingolipid, and depending on what kind of sugar we are attaching, whether it is a monosaccharide, then we will call it cerebroside. If it is a complex saccharide, then we will call it ganglioside. So now let us come back to the question and the sentence that was there. So the correct option should be A. Now let us complete the sentence. A cerebroside is a ceramide with a monosaccharide such as glucose or galactose. Just in the classification I have written, a cerebroside is a ceramide with a monosaccharide being attached. So yes, P was monosaccharide. Coming to the next part, whereas a ganglioside is a ceramide with a polar head group, that is a complex oligosaccharide. That I have written here, ganglioside, when we attach some complex sugar molecule or complex oligosaccharide, then that is our ganglioside. These cerebrosides and ganglioside are collectively known as glycosphingolipids because they are originally classification for sphingolipids, and as we are attaching sugar molecules, so tells their name, the glyco parts means sugar. That's why in general altogether cerebroside and gangliosides are called as glycosphingolipids. Okay, hope this is clear. Moving to the next one. Fine. The next question says, the given figure shows a schematic representation of the various types of membrane proteins. Identify the types of proteins labeled as X, Y and Z. Okay. So this is your membrane protein. This blue part that you are seeing, that is your membrane protein. And there are other proteins like this green one, this brown one, yellow one and the red one. Okay. These are the other proteins. You need to identify their type. These are the 4 options. Now let us see. First let's comes to X. So we can see that this X protein is sitting on the periphery of the membrane protein. This is the periphery, the outline of this membrane protein, this deep blue outline. This X protein is actually sitting outside or on the periphery. There is no part of X that is embedded within this membrane protein, or there is no anchor that is within this membrane protein. It is totally sitting outside on the periphery. That means X is nothing but a peripheral protein, right? Sorry. So as this yellow one, both of them, none of their part is inside the membrane protein. All of them are outside the or on the periphery of the membrane protein. So our X should be peripheral protein. Now let us come to Y. In case of Y, we can see that yes, there is some part of this protein which is present on the periphery, but it is also embedded within this blue square. That means this Y protein is actually embedded within the membrane protein, which implies that it is an integral part of the membrane protein. It is a structural part of this membrane protein because now it is fully embedded within the blue colored membrane protein. That means Y is our integral protein. Now let us look at Z. This red colored Z1, this red part is sitting on the periphery. No red part is inside the blue rectangle. That means the whole red part is outside. It is kind of looking like a peripheral protein, but there is a purple colored anchor type looking thing which is embedded in the membrane protein. Although this red part of the protein is not embedded, but there is a small part which is attached within the membrane protein. This kind of proteins are called lipid anchored because they are anchoring through a small portion. This whole protein is not embedded, but they are anchored to the inside of this membrane. That's why they are known as lipid anchored. Okay. So now we know. So this is our lipid anchored protein. So now we know X is peripheral, Y is integral and Z is anchored. So the correct answer option should be option number A. Okay. Moving to next one. This question state, identify the states as A and B given that the diagram represents the membrane fluidity and how it varies with temperature. Okay. So these states actually represents you the membrane fluidity. Now you have to identify which state is what. Now looking here, the polar head groups are same for both the cases. But in case A, you can clearly see that the fatty acids are highly arranged. They kind of look like crystalline planes, right? That means they are highly arranged state. That means, as I said, whenever there is very nice ordered arrangement, there will be no fluidity, there will be no passages to go between them because they are tightly packed, very nicely and tightly packed. That's why this state has less fluidity. We just discussed it in some other question. And this kind of state where highly ordered structure is maintained with less fluidity is called the gel state. And whenever you heat this kind of system, you basically increase the randomness in the system and thereby the ordered structure that you had earlier is no longer present in this case for B. You can already see now the fatty acids are kind of, or the lipids are actually randomly oriented. Nothing like what was in the gel state, which allows more fluidity to the membrane. And this kind of randomly oriented structures for bilipid membranes are known as liquid crystal. So what should be our correct option? A is our gel state and B is our liquid crystalline state. So the only option that matches it is option number D. So this was our correct answer. Moving to the next one. Match the following polar head groups with the appropriate glycerophospholipids. So to be able to answer this, again you should remember the classification of the lipids. So just in the previous slide or back to that, I discussed about the sphingolipids and their classification. Similarly, you have to remember the classification for glycerophospholipids as well. Fine. Now let us match what should be it. Previously just I discussed that in case of ganglioside, what you have? You have some kind of oligosaccharide. Sorry. In case of ganglioside, you have your sugar moiety attached with the OH group of sphingosine, that is a complex oligosaccharide. So our E matches with 4. Fine. Now in case of cerebroside, we said the OH group of sphingosine is attached with a monosaccharide or a simple sugar molecule. So our option number D matches with 2. In case of sphingomyelin, if you remember, in case of sphingomyelin, we actually have phosphated choline or phosphocholine. How does that look? In the last slide we said that we attach sugar molecule, we attach sugar molecule to get cerebroside or ganglioside. In another case, instead of sugar, we can attach it with a phosphate group initially, which can again attach with a choline moiety. So ultimately we will have a phosphocholine attached here. That particular classification is known as sphingomyelin. So our C matches with right hand side 1. Moving to the next one, phosphatidylcholine. What is phosphatidylcholine? Again, it is like a classification for glycerophospholipids. So in case of glycerol, you actually have 3 OH groups. Fine. Each of them you can attach with a fatty acid. If you attach, suppose R1 fatty acid here, R2 fatty acid here, but in case of C3, if you first attach a phosphate group followed by any polar alcohol like choline groups, then they are named after that alcohol that you are attaching. Up to this point, this is called phosphadiol. Up to this point, they will be known as phosphatidyl. Sorry, I have written the spelling wrong. Up to this point, they will be known as phosphatidyl. And after you attach any other polar head group to this phosphate group, then they will be named after that particular polar alcohol that you are choosing. In this case, for phosphatidylcholine, it is clearly can be understood from its name that for attaching we are using choline molecule. To phosphadiol we are actually attaching choline. So our option B matches with option number 6 at right hand side. And the first one is phosphadiol inositol. Again from the name, up to phosphadiol part it is same, but after this attaching of phosphate group, instead of choline, now we are attaching inositol. So this will be our A should be matched with 3. Phosphadiol inositol. So now only option that matches with that A3, B6, C is 1, D matches with 2 and E with 4. So after deducing that, our correct option should be option number A. Okay. Fine. So our next question says, plasma membranes are made up of what? For a many number of questions we're discussing about these plasma membranes or bilipid membranes. What are they made of? We know, first of all, we all know they are made of lipids, that is coming from your fatty acids, attaching fatty acids to different mother moieties, giving you lipids. They also contains carbohydrates, different sugar moieties, right? And again, they have many proteins embedded in their structure. So if I write it like this, these head groups generally contain different type of sugars or carbohydrates, etc. These tail, hydrophobic tail parts are made of different lipids or fatty acid, and the proteins generally remain embedded in the bilipid layers in different form to get more fluidity or to create different passages through the membrane or transportation. So our plasma membranes are actually a mixture of all these three: proteins, lipids and carbohydrates. Removing any of these three will not lead to a proper function of the lipid bilayers or bilayer membranes. Okay. So the correct option should be that plasma membranes are made of proteins, lipids and carbohydrates. Moving to next one. Fine. Identify the class of structures represented as the given molecule. Also state the backbone the molecule is involved. So you have to identify the backbone of the molecule as well as the classification. So first of all, let us look at the backbone. What is the structure? If you see here, this is C1, this is C2, this is C3. So we have 3 carbon atoms. This is the basic backbone structure I am just writing. C2, C3. With C1, we can see it is written as CH2OH. So let me write it down. CH2OH. Next, C2 is directly attached with the oxygen. So that will be CHOH. And the third one again is CH2OH. So C3 carbon we can write as CH2OH. So this is our C1, C2, C3. So this is the basic backbone structure of the molecule that is shown here. So what is this molecule? The backbone structure? It is nothing but our glycerol. So it has a glycerol backbone. We can already tell, and by identifying that you should already select the correct option. State. Let us look at the classification as well. So in this case, number 1 carbon and number 2 carbon, those two OH groups are attached with a fatty acid chain via ester bond formation, OCOR. This has also become OCOR in the structure. So these two sites are already occupied by fatty acids, and the third site has been occupied by a phosphate group. So what should be the name in that case? It is phosphatidyl or phosphatidyl backbone. Sorry, phosphatidyl or phosphatidyl, and this is a glycerol backbone. So the correct option for this question should be option number B. It is a phosphatidyl. The classification of this molecule is phosphatidyl and the backbone is a glycerol. Okay. Moving to the next one. So this is the classification. Assign the designation type in the nomenclature of the given two molecules. Okay. There is one thing wrong. The numbering should start from here Instead of 1 here, the numbering should start from here. 1, 2, 3, 4. This should be 5, 6, 7, 8, 9, 10. In this case, 10. Okay. Here also the numbering should have been started from 1, 2, 3, 4, 5, 6, 7, 8, 9, and this one should be 10. So these are the two type of nomenclature that is shown. You have to identify which one is what. So we already have discussed that in case of delta designation, we start from the COOH site. We start our marking from the COOH site. But in case of N designation, we start away from the COOH site. So in case of A, as we are starting from the COOH site, so A should be our delta designation, and B, as we are starting away from COOH group, B should be our N designation. So the correct option here should be A delta and B N designation. Okay. One second, excuse me. The next question states, which of the following best explains why the plasma membranes of cell exhibit a negative resting potential? Okay, which of the following best explain that why the plasma membranes of cell exhibit a negative resting potential? So you have to look at the potentials of these individual charges. We already know that in case of plasma membrane, there is a Na plus K plus transport along the plasma membranes, which results in a appropriate negative potential that is necessary for our body to function. So in case of in case of plasma membranes, if it is our plasma membranes, if it is inside the cell, just draw it like that, and if it's outside of the cell, that means it is extracellular. This is our cytoplasm or intracellular part. Okay. Now there is a potential that exists alongside this membrane due to the transportation of Na plus and K plus ions. Now which of this transformation gives rise to the resting potential? Now we already know that inside the cell K plus concentration is higher. That means the proteins or transport proteins that exist in these membrane sites actually favors to take K plus ions from outside and take them inside to keep a high concentration of K plus ion inside the cells. Whereas the concentration of Na plus ions are higher outside the cells. Okay. So let us look at the option. To maintain this potential, what you need? You will always need a greater concentration of K plus inside. That means the membrane should be, or the membrane is mostly permeable to K plus. If the membrane is mostly permeable to K plus only, then only the K plus concentration inside the cell will remain higher. And because now the K plus concentration is more inside and less outside the cell, so this concentration gradient will help in regulating a fixed concentration of K plus ions inside the cell. Suppose for functioning, I do not exactly remember the millimolar value for K plus concentration, but suppose say we need around 150 K plus ions inside. Suppose. Now, if the membrane is very much permeable to K plus ion, they can easily come inside. But whenever this number, suppose it is getting increased to 200, okay, because K plus ions are getting transported through ion channels, that means after some time it can happen that inside the cell the concentration of K plus ions are much more than what we actually require to maintain it to a proper potential. In that case, what the cell will do? Because there exists a concentration gradient as well. Inside there is higher K plus and outside there is low amount of K plus. Because of this concentration gradient, some amount of K plus will diffuse out of the membrane as well. So to do that again, this membrane should allow the passage of K plus ions inside and outside freely. So ultimately we came to the conclusion that to maintain the balance or resting potential, the membrane should be mostly permeable to K plus ion, and the K plus gradient, this concentration gradient, favors its diffusion out of the cell whenever it is required. Okay. So the correct option for question number 20 should be option number D. Okay. Let's just look to the next one. This question state, this is the representation for fluid mosaic model. Okay, the most acceptable model that exists for plasma membranes. Its main function is to separate the contents of the cell from the exterior. We all know that. The detailed representation of the model is shown above. Identify the unknown elements P, Q, R from the image. Okay. So this is the fluid mosaic model of this membrane. This is extracellular fluid and this is your cytoplasm. Fine. Now what do you need to identify? This blue thing termed as denoted here as P. This thing termed denoted as R, and the small yellow parts that are denoted as Q. So you already know, if we look at this P protein, you can already tell that it is embedded within the membrane structure. It is not only sitting on the periphery like these other ones. It is fully embedded within this membrane structure. That means it is an integral part of the membrane structure or the membrane protein as well. So this blue part is nothing but a integral protein. So our P is a integral protein. We can already tell. Now let us look at Q. This is Q, small yellow molecules. What are these? We already know that these are nothing but cholesterols. They exist in between the fatty acid chains of the phospholipids because you need some fluidity. As just we discussed, if your lipid bilayer is too much properly oriented, then there will be no passage left for transportation. That's why you need kinks or some other irregularity in their structure to have more fluidity in the lipid membranes so that the transportation can smoothly take place. Cholesterols are these small molecules which actually sits in these fatty acid parts of the lipids, thereby hampering the orderness that was previously there. Suppose this was initial structure of the bilipid membrane. Now if a small molecule comes and tries to interact with them and sits here, the structure will no longer remain in that such ordered form. It will try to become something like this to accommodate the small molecule, so that cholesterol now sit here freely. That means you are hampering the orderness, you are increasing the fluidity. Although that was not the question, we just needed to identify. So this small molecule that is shown here, which is increasing the fluidity of the membrane, is nothing but our cholesterol. So our Q is cholesterol. Next one is this R1. What is this? This also looks same like this blue one, almost same like this blue P1. Now P was an integral protein, but the only difference in case of R, if you look closely, you will notice that there is a hollow part. This is an intersection that you are seeing. If you look at it from the above, you will be able to see a hole. Say this is the polar head groups of the lipid bilayer membranes. We are just looking it from above of this bilipid layer. In that case, we will be able to notice that there is a hole through which molecules can pass through the membrane. That means it is acting as a channel for transportation. So this kind of protein, this is the only difference that exists between P and R. There is no hollow part in case of P, although it was embedded, but there was no channel through it a molecule can pass. But in case of R, it is embedded as well as it has a channel through which other molecules can pass through. That's why R will be known as a channel protein. Okay, or a protein channel. So after finding out all these things, the only option that matches is option number A, that is P is our integral protein, Q is cholesterol and R is protein channels. Okay, moving to the next one. What does B, C and D represents in the given figure? So this is your carrier protein. You have to add this yellow one that is shown in the diagram. This is your carrier protein. Then three different transport modes are shown here. You have to identify these three different modes written as B, C and D. So first of all, what are they? Based on how do they work, we generally classify these proteins that exist within this membrane bilayer as carrier protein, or we call them channel protein. So what is the difference between these two? Carrier protein is something that will, say, take up one molecule at a time, lead to some configurational change. It will take up one molecule, some any kind of substance, specific substances obviously. It will take it up, lead to some configurational change, carry it with yourself, and then pass it to the other side of the membrane. Say suppose this is one side of the membrane, this is another side of the membrane. So it takes up from this side of the membrane, leads to some configurational change, carries it through the hydrophobic part of the membrane and then releases it to the other side. These kind of proteins are called carrier proteins. Okay, so you have a control. They are taking one molecule at a time. But in case of channel protein, we can imagine it like a gate. When the gate is closed, no molecule can pass through. Now if you open the gate, then many molecules can get to the other side as long as the channel or the gateway is open. You can think it like these carrier proteins is very specific they kind of check your IDs. Before taking you through the gate, they check your ID, identify you, and then only transport it through you through the gate. But in case of channel proteins, you are not checking their IDs at all. You are just keeping it open for some time and many people are coming inside. You cannot control the flow of the molecules in that place. If you do not check their, suppose you are not checking their IDs and just opening the gates, then you will not be able to control the flow of the people. But in case of carrier proteins, as you are specifically seeing them, changing your configuration and then releasing it to the other side, it is happening in a very controlled way, or the flow is very controlled in case of carrier proteins. Now in this particular question, we are only talking about carrier proteins. During this transportation, many type of things can happen. First of all, the carrier protein can act in such a way that it will take the substance only from this side to the other side. No backward movement is allowed. You can only take the substance, take it inside or outside, any unidirectional movement, but you cannot go back. Okay. The other thing can happen, that is, it can take one substance, move it inside, take a different kind of substrate from the inside and transport it to outside. Okay, these two substances are different. A different substance is going inside, but another substance is coming outside. And in the last case, what can happen? It can take two different type of substances in the same direction, but you are not getting back. Okay. So these are the three types. How are they classified? The first one, where only a single substance is allowed to go through a single direction. This is known as uniport. C, where you can take two different type of substances. One can go inside and other will come outside. This is known as antiport. And the third one, where you can take up two different substances but can transport them to only a single direction, is known as symport. Okay. So our correct option should be option number B. Moving to the next one. So this is a mathematical question. I hope I would like all of you to solve it. So it is stating, consider a cell membrane with a potential difference across it, given that the delta mu value is minus 9000 joule per moles. You have to calculate the potential difference across the membrane and the R, F and temperature values are given. So what equation we need to consider for it? Let me write it down. We have to use delta U mu equals to RT ln. Suppose we are talking here about calcium 2 plus ion. Okay. Calcium 2 plus ion at V2 divided by calcium 2 plus ion at V1. Because we are talking about the transportation of ions, apart from their concentration gradient, there exists another thing, that is potential gradient, because they are charged species. So we should consider not only their concentration gradient but their charge gradient or potential difference across the membrane as well. So the next part of this equation will be ZF V2 minus V1. This part is coming due to your concentration gradient. This part is, second part is coming due to potential gradient. The detailed derivation of this equation is already discussed in the lecture session. So now let us come back to the mathematical problem. We have our R value 8.314. Temperature is 25 degrees centigrade, that means 298 Kelvin. Now the concentration of calcium 2 plus in V2, that is 10 millimolar, and the concentration of calcium 2 plus in V1 is 1000 millimolar, plus what our Z value is, the calcium, the charge of these ions is 2 plus, that means for calcium 2 plus Z is 2 because we have 2 plus charge here. So in place of Z we have to place 2. For F we will put 96500, and we need to calculate the potential difference, that is V2 minus V1. So all the values are given in your question. You just need to put them in the equation and you will get your V2 minus V1. If you solve the entire thing, then you will end up with a V2 minus V1 value of V2 minus V1 is 12.5 millivolt. Okay, just write down the equation and from the question fill the concentrations, Z values, F values, temperature, R value, everything, and you will get your potential difference V2 minus V1. Okay. Moving to the next one. This question states, given is the working of sodium potassium pump basically. Fill in for the unknown parameters X, Y, M and N. So what is it showing? This is your membrane protein. This pink one is your membrane. This grey colored one is your membrane protein that is transporting K plus ions and Na plus ions. The left side is your inside. This is cell, and the outside is, say suppose I am writing is a cytoplasm because it is inside cell, and it is our extracellular. We all know inside the cell K plus concentration is high. That's why it is transporting, because the protein always transports K plus from outside to inside. For Na plus, the concentration is higher on the outside because this membrane protein takes Na plus from the inside and keeps it outside of the membrane. Now you have to identify these things, that how many K plus gets transported and how many Na plus are transported at a single time. Now the protein that is responsible for this is also known as valinomycin. If you look at the structure, different type of structure, it exists in such a way. Suppose this is the hydrophobic part and this is the hydrophilic head groups. Just forget it. This cavity size made by the protein is as such that it can fit specifically a certain number of K plus ions or a certain number of Na plus ions. Now if I remember correctly, the radius for K plus ions was 1.33 angstrom, whereas for Na plus it was 0.001 0.98 angstrom. Now this protein needs to transport these ions as such that the concentrations are maintained as required. Also they do fit in the cavity. So from the look of it, you can already see as K plus is, the radius for K plus is higher, this same protein will not be able to fit more number of K plus molecule inside it. Suppose if it is X number of K plus and if it can fit, and if it can fit Y number of Na plus, any of them, because K plus is larger in size, obviously the number of X will be much less, that is number of K plus ion will be much less compared to Na plus, because Na plus is smaller in size, so it will be able to accommodate more number of Na plus ions inside. That is what happens as well. 3 Na plus are taken outside the cell and 2 K plus ions are transported inside the cell. So our X is 2 and our Y is 3. Okay. And what is M and N? This is the concentration of K plus and Na plus ion outside and inside. M is high because K plus remains high intracellular region. So M is high and Na plus remains high at outside region. So altogether M denotes a higher value. And N is low similarly because Na plus is lower in concentration inside the cell and K plus is lower in concentration outside the cell. So N represents a lower concentration of these ions. Okay. I think this was the end of this. Fine. Okay. This was the end of this today's questions. Okay, we will end our session now. You from next day we will start from week 8 lecture session question. The question will be related to structures of DNA and RNA, etc. Okay, we will start that from the next session. For now, I will stop.